So it's 9 o'clock. So it's time to start this webinar. So good morning, good afternoon, and good night. I understand that we have audience from several continents. For the reason, good morning, good afternoon, and good night. My name is Carlos Muscus. I am a member of the local organizing committee. And today I am replacing to Dr. Ivan Darío Vélez, who is the president of the Congress. So he's just attending activities related with the organization of this event. I want to welcome all of you to the sixth webinar. As you probably know, these are activities prior to the next Worldleish7 to be held in the city of Cartagena next year from August 1st to August 6th. So on a score, we hope to see all of you in the beautiful city of Cartagena. The organizing committee also want to thank to Dr. Elisa Cupolillo, who is our invited speaker, for accepting the invitation to give us this new webinar. Soon we will be informing the next webinar. So through Facebook, through Twitter, and also through our website. So, but this seminar, the next seminar, we expect to be before the end of the year. After this brief introduction, I want to invite my friend, Dr. Alexis Mendoza, who will act as moderator of this presentation and who will introduce Dr. Elisa Cupolillo. Thanks, Dr. Elisa, and thanks, Dr. Alexis, for accepting the invitation. So, go ahead. Alexis.
Okay, good morning, dear colleagues, friends, and general publics. Welcome to today's webinar. It is a great pleasure and honor to introduce Dr. Elisa Cupolillo, a great friend and scientist in the area of leishmaniasis, who will be our speaker today. Welcome, Elisa. Elisa is senior research at the Osvaldo Cruz Foundation, Fiocruz, head of the Leishmaniasis Research Laboratory, responsible for the reference service in leishmaniasis diagnosis and Leishmania species identification of the Brazilian Minister of Health and Pan American Health Organization, PAHO. Curator of the Leishmania collection for the Fiocruz Institute, expert in molecular systematic of Leishmania to reveal epidemiological pattern of leishmaniasis. Have several research projects with an extensive network of collaborators specialized in different aspects of leishmaniasis. Elisa's seminar is titled Species Diversity or Diversity of Name, Power Hiding Behind Leishmania Taxonomy. Please, Lisa, welcome. Go ahead.
Thank you, Alex. I will share my presentation first. How can I share this?
Yeah.
There is a button. There is a button at the bottom of the screen. So share the screen.
Share the screen. Yes.
And you select your file.
Mm-hmm. Is that shared?
Not yet.
No?
No.
Oh, not yet.
Yeah, it's strange. asking me for permission.
Luisa, can you give
Es
permission
solo
to
darle en el botón verde donde dice compartir. El icono de...
I'm doing
Ya.
it.
Elisa, did you push the green button in the bottom of the screen?
Yes, yes, I did. But it's not sharing.
Yes, it's not there.
No, no. Once more, let's see now.
it is?
Yeah,
Yes,
Yeah.
yes.
Could you put the presentation
yeah, in the
presentation mode?
Now it's okay, right?
Okay, thank you.
That's right.
So it's perfect.
Go ahead.
Okay, sorry. So let's start. Hello, everybody. Good morning, good afternoon, depends on the part of the world that you are. It's a pleasure to be here. I really want to thank Dr. Alex Mendonça Leon, a good friend that I met many years ago, and Professor Ivan Dario and Professor Carlos Muscos for inviting me to present this seminar as part of the next Worldleish7. It's a pleasure to be here with you, and I hope you can share good times next year also in Cartagena. And another thing that I wish is that everyone here that's attending this webinar, as well as your family and friends, are in good physical and mental health. We are living a very bad moment in the world, so I think it's the best thing that you can wish now. So the subject that Alex gave me, it's indeed a great challenge, and I think we have much more to reflect on and try together to contribute with taxonomic studies of Leishmania, a science of extreme importance, but somewhat neglected. We are missing taxonomists, not only for Leishmania, but for different organisms. And this is a big problem when you are discussing about biodiversity and mainly when you are discussing about pathogens. So how could there the advance in science itself without a correct classification of organisms? Is something that you must think on that to make good advances. Well, I chose to start with what we have most concrete and recent in terms of attempts to classify Leishmania parasites. Can you see the points? Yes? No? Oh.
Yes, we can. We can do.
Yes, good. So I will select to start by two recent published papers by the group of the Czech Republic. It's a group that it's heavily involved in taxonomical studies of trypanosomatids, including Leishmania. And in these two studies, they follow the International Commission on Zoological Nomenclature to try to deal with classification of Leishmania parasites. What's a good start, but also is something controversial when it comes to protozoal taxonomy, and we will discuss this a little. So here they clearly identify the type species of the genus Leishmania, which is well known to everyone, is Leishmania donovani, that was described by Laverhan and Mesnil, although these authors identified this parasite as Spiroplasma, and the creation of the genus Leishmania was made by Royce in 1908. So this is for sure the first revision in the nomenclature of the genus Leishmania, starting as Spiroplasma and now Leishmania, that is a well-defined, well-known, and valid genus. A mistake made in this first study that was corrected in the second was to consider the genus Leishmania as a synonym of Euleishmania. And this was correct based on the rules made by the International Commission on Zoological Nomenclature. And these two sections, Leishmania and Paraleishmania, were proposed as sections, and are names that not follow the taxonomic rules. So that's why cannot be considered a synonym of the genus Leishmania or of any other taxonomic rank. But these two groups are well supported by different studies conducted using different markers. Here are only examples using isoenzymes and using DNA, RNA polymerase, but many other markers can distinguish between Leishmania and Paraleishmania. So are not taxonomical groups, but this division is very concrete in these parasites. The two studies that we are following here were based on the new classification proposed by Espinosa and colleagues in 2018. And this was a new classification proposed, a new classification based on concatenated sequence of two genes, HSP70 and GAPD8. And also considering biological characteristics of the studied species. So they consider different members of the Leishmania family, but we will concentrate on those related to Leishmania, very closely related or belong to Leishmania genus. So here again, the two sections, Paraleishmania and Leishmania, very clear identified. But now the Paraleishmania, they proposed two different things. First, to name the Leishmania colombiensis, Leishmania hairy, and Leishmania equatorensis. In the case of colombiensis and equatorensis, were species described first as Leishmania viana. You see that it's a subgenus completely different from this group. So they proposed to name this Leishmania species as Andotrypanum, and also they proposed two new groups. One new genus, including two former Leishmania parasites, Leishmania ertige and Leishmania gini, and another group that belongs to a subgenus inside the Leishmania genus. So now the Leishmania genus is divided in two, is classified in four subgenus. Leishmania mundinia, that includes Leishmania martinicensis and Leishmania henriette and others. Leishmania martinicensis, it's important to say that's a cosmopolitan species, a species found in many parts of the world, in fact, in different hosts, and also Leishmania sauroleishmania, that's the Leishmania parasites from reptiles, and Leishmania viana and Leishmania leishmania. Those two represent the groups that include the well-known species of Leishmania, the most studied by different parameters, clinically talking, immunologically talking. So many studies are developed with these two groups because they are important human pathogens, of course. The idea of this classification in subgenus started many years ago, but But I think the milestone was the study published by Lanson Shaw in 1987, which presented the subgenus Viana, was the first time that they presented the subgenus Viana, and the classification in this subgenus, Leishmania, at that time, Leishmania, Viana, and Sauroleishmania, was based mainly, but not only, but mainly in the development of parasites in the gut of the insect vector. So, but this classification in subgenus is supported, very well supported by also by different markers. Here, very few examples, HSP70, I'm using that a lot because it's one marker very useful for Leishmania typing. And so HSP70, DNA and RNA polymerase, so showing not only the subgenus that I mentioned, but also the group named Leishmania that now belongs to the genus Endotrypanum following Spinoza and colleagues classification. So, but you still have it difficult to classify this group, to classify this species of Leishmania, and cytochrome oxidase 1, a mitochondrial gene, is the most commonly used barcode region for animals and also for some proteins, but it's not used yet for Leishmania. So there are many markers already proposed, but we are still looking for some that can give better answer, can discriminate better the species that are validated by different characteristics. And the study from our group indicated this as a good target for Leishmania identification. We didn't publish this yet, but you can see a good barcode gap so that can distinguish not only those high rank taxonomic ranks like the subgenus Viania, Leishmania, and Sauroleishmania, the new genus Forsythia, the species belong to the Endotrypanum species. But here I also want to highlight that this region allows identification of most of Leishmania species. I will talk about this a little later. And mainly those species belong to the Viania and Leishmania genus that, as I mentioned, represent the most relevant human pathogens. So, but if you are making good progress on the classification of Leishmania parasites in higher taxonomic levels, we still have some questions, like how many Leishmania species were described so far? How many represent valid taxons? So there are many works saying that you have 20, more than 20, more than 30, 35. So you don't know very well how many species were described and how many represent valid taxon, because some are not very well studied. A precise answer is impossible yet, but it seems that the last species described, it's as far as I know, was Leishmania orientalis, that belongs to the Mundinia subgenus, and the classification in species complex and subspecies were also proposed for this group of parasites, but I'm not going through exactly, not in deep on this right now. It's many more things to discuss, and the time is not, it's a little bit short. So I think here we have a good start to think on Leishmania species concept. It is an old paper published by Michel Tibaran, and something that he mentioned, I think is very interesting to start thinking. First, he says that species are not mere fantasies. It's possible to identify evolutionary entities that deserve to be attributed the name species and that are relevant to medical researchers and decision makers. This is very important when you are talking about a human pathogen. But for those who had the pleasure to attend Professor Jeffrey Shaw's seminar, it was the last one before this one, it is important to remember what he says about the ecological importance of Leishmania species, and that we know little about those that are not human pathogens, but that are very relevant, and not only when you discuss about biodiversity, but also about the impact of causing human disease in any moment, and depends on the disturbed in the environment made by humans. You know that this is possible for many Leishmania parasites. So the two principal questions that I have in my mind, and is something that I'm trying to discuss with different colleagues, and you are trying to contribute a little, is it possible to have a definition for Leishmania species, looking for fungi, for bacteria, thinking other microorganisms? Maybe we can find a way to have a better definition for Leishmania species. And another point, should we use a plurality of equally correct definitions? So we know that Leishmania parasites, most of the species, if not all, were described based on ecological, epidemiological, and biological characteristics. These characters are the base for Leishmania taxonomy when we are discussing about species. Transmission cycle, biological behavior, not only experimental vertebrate laboratory animals, but also in the vectors, clinical manifestation, geographic distribution, but all these aspects have some controversial, and we must be careful when you are discussing about this. And also, it's not easy to follow all these characteristics to describe the species. It seems that these criteria are not stable enough to define species, but what's important to say that many molecular data are validating most of the species already named. So there are some points that it's important to consider when you are discussing about taxonomy, about classification, about the description of new species. First, critical elements of evolution and ecology were elucidated for a minority of those species that have been named, something that very well discussed by Professor Jeffrey Shaw. Of course, his talk was excellent, and he discussed a lot about this evolution, the ecology of Leishmania, and the fact that you know very little about some species. The association with human infections drive the discovery of new parasites, new species or not. So we are seeing just the top of the iceberg. You are not seeing too much species that are circulating in the nature, in the natural reservoir, but most of them were those associated with the human disease. The emergence of the disease will be crucial, of course, because without it, the parasite will hardly be seen. It's not easy to do field research, and it's being harder and harder to do that. And this is represented by the few samples available that came from sandflies, from natural reservoirs, and it's not like in the past, unfortunately. It's estimated that no more than 10% of global pathogens have been documented. So it's very few. You have much more pathogens that are circulating in the world, and very few are documented. And this is for sure the case of Leishmania. And Leishmania is basically considered as a wild parasite. So if you don't look for the reservoirs, it's hard to understand the diversity of this parasite. And its association to human is occasional. And it's the part that you are looking for. So, but how can you classify Leishmania taxonomies? As splitters that like to divide it in many groups to describe the many species, considering different characters, or as a lumpers that want to look only for one marker and put together species that we know that are biologically distinct. For example, when you use HSP70, it's not so easy to distinguish between Leishmania amazonensis and Leishmania mexicana, but you know that they are very distinct species. This is probably indicated that we still do not have one marker that allows to identify all species described, and not that many species are not valid. So you cannot use only one marker to validate the species that can be distinguished by different biological, even that biological characters are changing, you cannot validate that only using one marker. Maybe this is the point, but you need one marker, a barcode marker, to identify fast, easy in the laboratories. So in any case, no name, no information. This is classic in taxonomy. It's not an idea of myself, but No name, no information, wrong name, wrong information. And this is very serious when you are dealing with a pathogen, because many information can be achieved, but associated to wrong parasites, to wrong species. An aspect that it is important that maybe makes difficult to find the markers is the fact that multi-lox enzyme electrophoresis, isoenzymes, is still considered the gold standard. Many authors will argue that no, it's not the gold standard anymore. But when you describe new markers, you always, the slide's not very good, but the column here, when they say species here and here and here, all the species were considered in this study, looking for the results based on isoenzymes. So it's not easy to propose new things when you are trying to correlate with something that maybe is not good to define species, but to type them when you have the appropriated set of enzymes. So a multi-lox enzyme electrophoresis is a good approach for Leishmania species typing. Yes, I think so, but not to name new species. For example, there are many drawbacks, many problems with isoenzymes, but I would like to highlight two aspects. First, that several Leishmania species like Brasiliensis and knife present a distinct profile for several enzymes, but they all belong to the same species. Here's an example of many zymodemes belong to Leishmania brasiliensis. And from the other side, distinct species present difference in only one enzyme. This is the case of L. archibald, Leishmania archibald, comparing to Donovan, Leishmania donovani, and also Leishmania peruviana represented here, that can be distinguished when using isoenzymes only by the mobility in the mannose phosphate isomerase. So, but the case of L. peruviana is quite emblematic. I'm sorry, I'm focusing more in the New World species because are those that I have more experience, so I feel more comfortable to talk about them, but it's not exclusively for the New World. Out case that here I could speak about Donovan and Archibald, for example, but I prefer to show the case of L. peruviana is quite emblematic. For a long time, the validity of this species was questioned if Leishmania peruviana was a distinct species from Brasiliensis or not. They proposed to be a subspecies and different classification or only a synonym because of the low level of difference when you look for the isoenzymes. But you know that these species have distinct geographic circulation, present distinct clinical manifestation, and recently it was published a study by the group from the Institute of Tropical Medicine in Antwerp, conducted by the group of Jean-Claude Dujardin, and analyzing the completely nuclear and mitochondrial genome. It was possible to clearly define the two groups, L. brasiliensis and L. peruviana. So there are many other interesting things showing the ecological divergence and the hybridization of neotropical Leishmania parasites, focusing in Leishmania brasiliensis and Leishmania peruviana from Peru in this study. But here what I want to show that the complete genome shows that what was demonstrated by the geography, the biogeography, and the transmission cycle and clinical manifestation was corroborated by the genome, whole genomic analysis. The same for Leishmania chagas and Leishmania infantum. For many years, this was questioned if Chagas was a species different from Leishmania infantum. Some authors consider Chagas as distinct species. Some authors did not consider. This is a long history, starting in the early 80s. But in this recent published paper by our group, we demonstrated the process of colonization and genetic diversification of Leishmania infantum in the Americas. So forming only one group represented here by samples, strains maybe. I don't know what a strain is for Leishmania. Alex asked me this last Friday. And sorry, Alex, but I don't have the answer for that. And I think something that you must think together about. But here we have, I will call strains, different Leishmania infantum strains from both New World and Old World, and all representing the same species. And so I think now it's very well defined that Leishmania chagas is a synonym of Leishmania infantum, but something that I start thinking, I mean, I not start to think exactly that, but I pay more attention. It was during the talk by Professor Jeffrey Shaw, when he mentioned about the subspecies for Leishmania chagas, being a subspecies of Leishmania infantum. And maybe it's possible to consider chagas a subspecies of L. infantum. It's another thing that you must think. And here I brought a concept that maybe is relevant to discuss this. A subspecies is an aggregate of phenotypically similar population of a species inhabiting a geographic subdivision of the range of that species and differing taxonomically from other populations of that species. So maybe you must start thinking about subspecies again in Leishmania, but very carefully to avoid more confusion. And maybe also this is the case for Leishmania brasiliensis. Here is a study performed using microsatellite, analyzing many Leishmania viana strains from different species, all the species circulating in Brazil, but many Leishmania guianensis, represented here by this population in green, and Leishmania brasiliensis that belongs to two distinct populations. Population 1 in red, that is dispersed in the Atlantic coast, mainly in the Atlantic coast, and population blue, population 3, sorry, in blue, that also comprise other Leishmania viana species, like Leishmania night, Leishmania lensoni, and are restricted mainly to the north of Brazil. So you can observe also in some other parts, but cannot guarantee that these represent autochthonous parasites, but they are mainly circulating in this region. However, population 3 can be subdivided in the species that we analyze, like knife, tingensis, lensoni, but also Leishmania brasiliensis can be subdivided in population 3A and 3B. Here, different groups of L. brasiliensis. So there are distinct populations of L. brasiliensis that people that works in the north, not only with the patients, with the clinical, with treatment, but also with the ecology of the parasite, can discuss this very well, showing differences between L. brasiliensis from the eastern and the western Amazon, for example. And maybe a coincidence, and I don't think it's a coincidence, but a process of coevolution, where we observe the population 1, Leishmania guianensis, the green circle, and population 3, the blue, doesn't matter if it's L. brasiliensis or other species. We also observe the presence of Leishmania RNA virus. So it seems that there is overlapping the circulation of some Leishmania population and the presence of this endosymbiotic virus. The role of Leishmania RNA virus 1 in the clinical outcome of Leishmaniasis has been extensively explored, showing not only a possible association with the mucocutaneous form, but also with the treatment outcome. I'm not going through this, and I think they should deserve a specific seminar, but what I want to show that when we analyzed the sequence of Leishmania RNA virus 1, here are the sequence of the virus, we can correlate this with the Leishmania species infected by them. So here is the virus infecting Leishmania shawi. In this group, only virus infecting Leishmania guianensis, and in this last group, only virus infecting Leishmania brasiliensis. And also another important thing that some subgroups correlates with the geographic origin, the geographic dispersion of the parasite. For example, here, there are parasites from Leishmania brasiliensis from Bolivia, and in this group here, Leishmania brasiliensis from Brazil. So it seems that there is a coevolution occurring between Leishmania RNA virus and the RNA virus and the Leishmania species. So you are almost at the end of the presentation, and you need to think on solutions. You need to think on species definition, standardized data acquisition, use of molecular data, and name stability. I would like to reinforce something that was published in the paper that described the Leishmania waltoni. It's a Leishmania waltoni, is a Leishmania from the Leishmania subgenus, closely related to Leishmania mexicana that was found in the Dominican Republic. And the authors mentioned something very important. In the present genomic era, it has been suggested that a bacterial species should be a biologically meaningful cluster. For obligatory pathogens, the ecological niches are very restricted and result in detectable clusters. This is perhaps a good start point for us to begin examining the species concept for Leishmania. So I think I agree with them that this is a good start point. And what you must think also is the two big problems that you have to solve. First, no more important than the second, is the question of the analyzed samples. Many studies use few samples, sometimes just one representing each species. And also different studies employ distinct strains, limiting the comparison of results. Another important aspect is the use of different approaches. While you should be looking to have a standardized protocol, not only to contribute to the description of species, but in the quick and accurate identification of those species, something very relevant for the clinicians. So I cannot finish this talk without saying that although we are in the era of genetic sequencing, independent of isolation, cultivation of parasites, we cannot disregard the great value of biological collections, and these have increasingly taken a back seat, unfortunately. It's clear that the effort to isolate and cultivate the parasite has decreased over the years. Currently, even though there are several repositories around the world, repositories of Leishmania strains, there are only 2 registered in the World Director of Culture Collections. Is the one here in Fiocruz, that I am the actual curator, and the other one in Montpellier, that Patrick Bastian is, I think, is still the curator there. So ATCC also is an important collection, but for Leishmania, in fact, there is many problems with this collection, in my opinion. Here, only few examples. You see that they are providing Leishmania donovania, that was deposited as Leishmania chagazi. So you don't know if the problem was only the nomenclature during the deposit or something missed here, mislabeling maybe. Another problem, there are many others. I only select 3 examples. Here is deposit as Leishmania donovani Chagas, probably using older nomenclature, and now is provided as Leishmania Chagas. And here for me is the most serious problem that this was deposited as Leishmania brasiliensis Viana and is being provided as Leishmania mexicana. So can you imagine working with this? Think that it's Leishmania mexicana, but in fact it's Leishmania brasiliensis or something changed. So it's not represent, you don't know where this sample is from probably. So to finish, I would like to propose a worldwide network that can share Leishmania cultures, information on these parasites, discuss and share standardized protocols that can be used in different types of laboratory dealing with Leishmaniasis. Important for the routine of each of these laboratories, important for the routine also when you are dealing with patients. We will also have the opportunity to perform a broad genomic analysis and use this not only for species identification, but to perform phylogenetic inference and make good and appropriate taxonomic revision. So hoping one day and very soon we can do that, working together, collaborate among us. So I would like to thank you, the audience, again, Alexis, Professor Ivan Dario, and Professor Carlos Muscos, and hope to see you in Cartagena, still alive with health, mental and physical health. Thank you.
Thank you, Elisa. It's an excellent conference that put in the desk the main problem of taxonomy of Leishmania. Well, now I invite to the colleague and to the public to put the question in. One thing is using the yellow hand or put it in the screen. The question. I have some comment here that, for example, Dr. Ricardo Goroy, hello, Elisa and colleague. I would like to thank the organizer for the event and the opportunity to participate. Elisa, thanks for the excellent presentation. Would you like to comment a bit further on the role of the genomic approach on Leishmania phylogenetic? This is one question.
Yes. Oh, thank you, Ricardo. Ricardo is a good friend that we work together in Rondônia. So we discuss a lot about this. So there are many studies on phylogenomics of Leishmania, but in fact, I don't think you already have a nice picture because you are working with limited set of sample. So you already have some clues about the evolution, like the Professor Jeffrey Shaw mentioned about that, the origin of the parasites, but it's still very difficult to discuss that when you don't represent the diverse. That's why I'm claiming people to collaborate and to put more samples together, to analyze together, a great effort to make a root work looking for samples from everywhere, from different parts. So, Ricardo, sorry, but I prefer not go through the phylogenomics studies because I think they are still very, very limited, although they are only showing what we already demonstrated with different markers. And sampling is a problem in these studies because they are only repeating very few advances were made so far. But I think the best works are those two, not only those, there are some with Leishmania donovani as well, and with other species. When you look different samples and do the phylogenomics of a specific group, but what is lacking is the phylogenomics of the whole genus, but using appropriated sampling.
Okay, there is a comment of our friend Patrick Bastian. He said that the term strain has been defined long ago by WHO. A strain is an isolate which has been adapted to in vitro cultivation. Hence, for this fact may differ for the original isolate. Any comment?
Yes, thanks, Patrick. Nice to see you here. Not see exactly, but to have you present here. Yes, I agree with him. This was the definition, and but I don't know. So this means that each isolate that goes adapted to the culture will have one strength. So it's not the same for other microorganisms. So you don't have any specific characteristic. I cannot, for example, many people come to the collection asking, I would like to type this to see if this represents Leishmania infantum PP75. Leishmania major LV39. You don't have markers for that, unfortunately. You don't have markers for that. But I agree. I call them strains, like Patrick said, but we don't have markers to discriminate them, not yet. Not that I know, but maybe Patrick has some clues about that, and I would love to hear that.
See, there is another comment of Chaden Kamali. Nice talk. How unique are the taxonomies in revision of the system, and how far are we from reaching a global taxonomy system for all of us to use? It's a comment.
Yeah. Hi, Shaden. I think you are very far from this. If you don't collaborate, it will be hard to get this global taxonomy that everybody can use. And if you look at the different papers, mainly those that are not dealing with identification, with taxonomy, many errors, taxonomic errors, wrong names, that were not writing the right way, so it's a problem. But I believe that soon you have this big network. It's not easy at the moment to share pathogens, to share biological samples. And in Brazil, for example, there are restricted laws that are almost it's difficult to share the samples. But you can share protocols. You can train people in the different labs, and you can try to do the same thing in different labs. And when you do service, for example, to do the whole genomic analysis, you can use service. So it's easy to send the DNA than the parasite alive. So I think you are close to that, but far from to have a final taxonomical picture. Actually, taxonomy is a sign that's multiplying every time. It's not a static science.
Exactly, right. Well, there is a Daniel Jeffers. Elisa, can you comment on hybridization in Guyana?
Yes, hi, Daniel. Daniel is also another good colleague that we are working together with some Leishmania species, mainly on Leishmania infantum. Yes, not only in Leishmania viana, but the paper that I showed looking for Leishmania peruviana demonstrated that the possibility of hybridization in natural parasites, and not only in the nuclear level, but also mitochondrial level. So what's really important. But in Viana, there are not that I know, experimental conducted showing hybridization, only the detection of natural hybrids. So we don't know yet if this is occurring in the nature of, if this represent old parasites, old lineages that were maintained by clonality. So it's something that you must study better, but there are some natural hybrids in Leishmania viana, many of them. For example, between different species, Leishmania brasiliensis and Leishmania guianensis, Brasiliensis and peruviana, Brasiliensis and Lensoni, Brasiliensis and knife. So it seems that something that occurs, but you need to know if this is an old event or if it's occurring in the nature, not very common, but it's occurring. It seems that it's occurring based on the reports for other Leishmania species.
Right. Mariana Boytet asks you, should we rediscuss all the issues in Leishmania taxonomy, including the species definition, marker, etc., based on whole genome sequencing data? Or the problem is not just method resolution, it's the agreement between researchers?
I think both. Hi, Mariana. Mariana is from my lab. We discussed this a lot. So what I think it should be better, but maybe this is a dream, is to have the whole genome for the different species and many representative of each species, and go back. With this genome, we can find the right markers for species identification. And maybe you can create an algorithm that can be used for species identifications. Depends on the region, depends on the epidemiology, or depends on the order. Maybe from the genome to the back. This is a dream, but I don't think it's impossible.
Well, here is Patricia Quaresma from Brazil. The question is about the recent report by Pitbull's group demonstrating that Cupolillo is capable of transmitting L. mundinia. What may be the impact of this finding on the taxonomy of Leishmania?
I don't know if in the taxonomy if this is affecting the taxonomy, but of course the evolution, and in some instances in the taxonomy, when the parasite has, the protozoa has the possibility of infecting several reservoirs, several hosts, insects, and of course the pressure, the biological pressure, the selective pressure is higher. And this will probably make the evolution faster for these parasites. And this will have an impact in the evolution and maybe in the taxonomy. But the mundinia is a very interesting group because it's so widespread and is in fact not only different vectors, but also different animal, vertebrate animals.
Okay. Well, there is a Andre Rocket. For sure, a standardized protocol for species identification is crucial, but don't you think that a nomenclature consensus will also be valuable?
Yeah, for sure. Yeah. Completely agree. Yeah.
Totally agree with that, right?
Complete, yeah, we must have a consensus in the nomenclature. There are different things, yeah, because nomenclature is one thing, taxonomy is another, classification, so there are an overlap among them, but different things. But of course, yeah, I completely agree. Another dream.
Right. Maxi de los Santos, what is the importance of taxonomy in terms of treatment? Because physicians use antimonial since more than 60 years ago for the majority of Leishmania species.
Yes, he's right, but we know that the same treatment scheme is used for the different Desameridae species. You don't perform species identification for the clinical conduction. That's true, but you are not sure that this is the best model. We know that there are some differences in the treatment, and you don't know very well why, if it's this represent failure of the patient respond to the treatment, of a resistance of the parasites, and you don't have good studies to associate it with different species. But what you know that the different species do not respond exactly equally to the different treatments available, and more studies are needed for sure.
Right. But our friends, Alicia Ponte Sucre from Venezuela, said, "Dear Alicia, many thanks for his beautiful and informative conference that emphasize the value of Leishmania typing. Does what you have presented mean that a thorough revision of the biological data based in different strains and species should be revisited?"
Sorry, I did not understand, Alex. Can you repeat?
Well, yes. The question is if you does what you have presented mean that a thorough revision of the biological data based in different strains and species should be revisited? In other words, it is necessary to revise different data, biological, in general taxonomy, to be defined strain and species.
Yeah, this is like the polyphasic taxonomy that it should be good to do, but it's very difficult. It's very expensive and very difficult to perform. I think that to describe a new species, yes, this is very important to be conducted. But, and you know that some species are being described not following all these approaches. But for identification, it's impossible. Too much work and very, very expensive.
Well, there is
But hi, Alicia, nice to see you here also. I miss you. Good friends, old friends.
Patricia Cuervo said, "Excellent presentation, Elisa. Thank you so much. As we recently showed that deep proteomics can distinguish very close Leishmania species, I would like you to comment on the potential of deep proteomics of taxonomy study."
Yes, this is a very good question. Yeah, Patricia published a study showing that by proteome they could separate. Actually, I'm a collaborator for this paper. Separate closely related species like Leishmania panamensis, Leishmania guianensis, and Leishmania brasiliensis. The problem that, again, we need to, it's maybe this can be a very good marker, but you need to do a broad study looking for different strains of each of these species, and then select those proteins that can discriminate among them. It's a challenge again, Patricia. Yes, I agree with you, and it's a nice work, but it's a challenge because it's not so easy and quite expensive to perform the proteomic analysis of a large number of strains representing the diversity of each species. But without that, you cannot guarantee that you have a specific marker.
Right. Sydney's asks, how is the level of genetic diversity of L. infantum in Brazil? Is it significantly high or not?
No, it's very homogeneous. We can detect some different population. For example, we perform a study using microsatellites that you described three important populations, and one very associated with the transmission cycle, including Lutzomia cruzi and not Lutzomia longe palpes only in the transmission cycle. But even though that there is some populations, it's a very homogeneous. And also recent paper published by Juan David Ramirez in Colombia also show that the Colombian strains are very homogeneous as well.
It's okay. Claribel Murillo asks, is there any evidence of the influence of the culture condition milieu in the taxonomy classification outcome?
Not in the taxonomy, no, it's very stable. But we know that for genomic studies, yes, the medium, the number of passages, in vitro passage, all this can influence the genomic organization of Leishmania. There are several recent studies approaching that and very interesting results, but not in the taxonomy. What can occur is mixed infections. And you can detect one parasite, one species at the beginning of the identification, but if you cultivate that for so long, you can find another thing. And not is a mislabeling or contamination. Sometimes mixed infection in the reservoir, in the patient, and that is selected during in vitro cultivation. This is quite common in the Amazon region, for example, where many species are circulating.
That's okay. Okay, CD Hearing asks, thank you for the interesting presentation of the Leishmania taxonomy. Assuming phylogenetic and maybe coevolution between Leishmania virus and Leishmania brasiliensis, what implication can such a phylogenetic evolutionary pattern have on contemporary speciation process?
Yeah, that's a question that we also have. I don't have the answer yet, but what's interesting that many Brazilienses, Leishmania brasiliensis, circulate in Brazil, for example, in the Atlantic coast, they do not present the endosymbiotic virus. So you need to understand if this is acquired resistance to the infection. But this is something that you start studying right now, and in collaboration with Frederick and Jean-Claude Dujardin in Belgium. So hope soon we'll have a good answer for that. But for this, we are sequencing not only the virus, but the same parasite that the virus is infecting, to do the phylogenomic of both. Sorry, I don't have the answer. But maybe someone here in the audience can help with this.
It's a different question, right? Marianne Boyd, excellent presentation, Elisa, thank you. My question is, should we rediscuss all the issues in Leishmania taxonomy, including the species definition, marker, etc.?
You already did this question.
Yes, okay. Sorry, but because
no,
there is
okay.
so many. This is our friend Jeff Cho. Congratulations on your talk, Elisa. Understanding the evolution is the only way that makes sense of understanding group and how they are evolving today. I agree with you that we need a standardized method, and that a barcode is an option. Do you think that the barcode must be based on cytochrome?
At the moment, yes, to say something to my professor, Jeffrey Shaw. He's my good professor, and it's an honor to be here with him. But at the moment, yes, I think it's the best mark barcode, the barcode using mitochondrial genes. It was published a recent paper in Memórias do Instituto Oswaldo Cruz by the group from Panama, showing the use of cytochrome B. That also seems to be good. And the results in our lab is showing that cytochrome oxidase 1 also can be a good approach. Yes, I think so. You still need to find this, but maybe go from the genome to find the best barcode marker will be better. Thank you, Professor. Nice to see you.
Well, a really interesting discussion. I think that the time is close to finish, but I only one comment, Elisa. There is, you mentioned different molecular marker using in the identification, but in your experience, for example, if you need to recommend to some people one of them or some of them, what do you think that is more properly molecular marker?
Depends on the question, depends on the region, depends on the suspicions that you have. So maybe starting by, now it's not published yet, but in the lab, we are using cytochrome oxidase 1. And always isoenzymes, you need to do that because of the collection, you still need to do the isoenzymes when we have the isolate, the culture. But cytochrome oxidase 1 seems to be very good. But sometimes you need to go for a pipeline, like starting by HSP70, go to the ITS, and then for some multi-local sequence typing, and looking for the genes for codifying for 6PG, GXP, MPI, and so on. So depends on the question, depends on the region, and depends on your suspicions. There is not only one marker.
Right. This is a good answer because you know, many people that work with Leishmania usually isolate one strain and then try to characterize and sometimes use a reference strain that usually when you check some market, there is no reference strain. And then it's too complicated. Well, Elisa, thank you very much for this opportunity to talk about taxonomy in general and application. Probably, I think that it is important to mention that the taxonomy problem is in the desk, and then we need to continue the discussion about that. Thank you for your participation. I hope to see you again very soon. And thank you for the public in general and colleague and other person. I don't know if Dr. Carlos Musco want to say something.
I just want to say on behalf of the organization committee, I want to thank again Dr. Elisa Cupolillo and also Dr. Leonce Mendoza for this interesting webinar. We have many comments in the chat about the excellent presentation. Thank you also to the audience, and hope to see all of you in the next webinar, as I mentioned before, at the end of the year. Thank you, Dr. Elisa, and have a good day, good night, and good morning.
Yeah,
many people all over the world. Thank you.
You too. Thank you, Dr. Carlos. Thank you, Alexis. Nice to meet you again. Bye.
Bye, everybody, and thank you.
Bye. Thank you, everybody.
Bye. Okay, we have to finish. We have completed.
Yeah, we have to finish. Thank you.